Okay, so have you ever wondered how your body fights off those nasty bugs that are constantly trying to invade and make you sick?
I mean, it's kind of a miracle, right, that we aren't sick all the time, given how many microbes are out there?
Right. It's pretty amazing. And we know there's this whole system in our bodies called the immune system that's our constant protector.
Totally. But today we're not just talking about any old immune response. We're diving deep into the fascinating world of adaptive immunity.
Yeah, adaptive immunity is like having your own personal bodyguard that learns the enemy's secrets and develops a super specific plan to take them down.
It's the next level of defense.
Exactly. So to get this right, we've been reading up on a chapter from a microbiology textbook.
The real deal with all the science-why details.
Yeah, we're talking specificity, memory tolerance, B cells, T cells, antibodies, this thing called the MHC, the major histocompatibility complex.
We're going to break it all down and make sure you walk away with a solid understanding of how this incredibly complex system works.
And hopefully some of those aha moments where you're like, wow, that's really cool.
Definitely. So right off the bat, the textbook emphasizes that adaptive immunity works hand in hand with the innate immune system.
Which is like your body's first line of defense.
Exactly. The innate system is quick and dirty. It's like a security guard who's just there to stop anyone from getting in, no matter who they are.
But it's not always enough, right?
Right. Sometimes you need a more specialized response, and that's where adaptive immunity steps in. It targets specific invaders and learns how to fight them more effectively over time.
So let's talk about specificity. The book says that B and T lymphocytes, those are white blood cells that are key players in adaptive immunity. They have these receptors called BCRs on B cells and TCRs on T cells. And these receptors are incredibly picky. Each one is designed to recognize only one specific type of antigen.
Like a lock and key.
Yeah, exactly. So these antigens are basically molecular markers that are found on the surface of pathogens, like a name tag for each bad guy.
And our immune system can create a unique receptor, a specific detective for each of those antigens.
That's incredible. So each pathogen might have multiple antigens on its surface, like a bunch of different wanted posters.
Exactly. And our body can create a matching lymphocyte for each one. It's like having a specialized team of detectives, each assigned to track down a particular criminal.
Makes sense.
Yeah.
Now, this whole idea of memory is super fascinating.
Yeah.
The textbook explains that the first time your body encounters a specific antigen, it's called the primary response. And it takes some time to build up defenses.
Yeah, like a first encounter with a new foe. It takes a while to figure out their weaknesses.
Right, but during that primary response, the body also creates memory cells. These cells are like long-lived veterans who remember the specific antigen and know how to fight it effectively.
So the next time the body encounters that same antigen, it's like those veterans jump into action immediately.
And they mount a much faster and stronger attack, called the secondary response. The book even mentions it can be 10 times stronger or even more.
That's why vaccines are so effective. They basically introduce a harmless version of the antigen.
Giving your immune system a chance to create those memory cells without actually getting sick.
Exactly. It's like a training exercise for your immune system.
So when the real pathogen shows up, your body's already prepared to fight it off.
Precisely. Now, another key principle of adaptive immunity is tolerance.
This is where things get really sophisticated, because it means our immune system can distinguish between friend and foe.
Between self and non-self.
Exactly. It knows not to attack our own healthy tissues while still targeting those foreign invaders.
The book points out that when this tolerance breaks down, that's when we get autoimmune diseases.
And which is when the immune system mistakenly attacks the body's own cells as if they were enemies.
Yeah, it can be a really serious problem, so it's essential that the immune system gets this right.
Absolutely. So how does the body actually train these T cells to be both effective fighters and respectful of our own cells?
Well, the textbook dives into T cell selection, which happens in the thymus. This is a primary lymphoid organ, basically a school for T cells.
Kind of like boot camp for the immune system.
Exactly, and in the thymus, T cells undergo a rigorous two-stage selection process. The first stage is called positive selection.
Where the T cells that can recognize self-MHC molecules are allowed to survive.
It's like a basic compatibility test. If they can't even recognize the body's own MHC molecules, they're useless and get eliminated.
So positive selection ensures that the surviving T cells have the basic tools to function in the immune system.
Right, but then comes negative selection, which is even more intense.
This is where the T cells that bind too strongly to self-antigens are eliminated.
It's like weeding out the potential troublemakers, the ones that might attack the body's own tissues.
This process is called clonal deletion, and it's crucial for preventing autoimmunity.
It's like an internal affairs division, making sure that only the good guys get through.
The book mentions that an incredible 95 to 98% of immature T cells don't even make it through this whole selection process.
That's a huge number. It just shows how important it is to get rid of any potentially self-reactive T cells.
Wow, so the survivors, the T cells that have passed both positive and negative selection, they graduate from the thymus and move on to the secondary lymphoid organs like the spleen and lymph nodes.
Where they can encounter foreign antigens and put their training to good use.
So what about B cells? They also need to learn tolerance, right?
Absolutely. B cells undergo their own selection process, but it happens in the bone marrow.
Another primary lymphoid organ.
And it's similar to T cell selection in that it involves clonal deletion, getting rid of the B cells that recognize self-antigens too strongly.
But there's also another mechanism called clonal anergy.
Which is a bit different. Instead of being eliminated, these B cells become unresponsive to the self-antigens.
So they're still there, but they're basically silenced.
Exactly. It's like putting them on probation. They're not allowed to mount a full immune response.
So both clonal deletion and clonal anergy are important for preventing B cells from attacking the body's own tissues.
Right. It's a multi-layered system of checks and balances.
Now, when a B cell actually encounters a foreign antigen that it recognizes, it gets activated and starts to proliferate or make copies of itself.
And it differentiates into plasma cells, which are the antibody factories.
And also those memory B cells we talked about earlier.
Exactly. So the immune system is prepared for future encounters with the same pathogen.
The book makes a distinction between T-independent antigens and T-dependent antigens.
T-independent antigens are like the simpler bad guys. They can activate B cells directly without any help from T cells.
But the response is usually weaker, and it doesn't create those long-lived memory cells.
It's like a quick hit-and-run attack.
Whereas T-dependent antigens, which are usually more complex, require the involvement of T helper cells to fully activate the B cells.
And this leads to a much more robust and long-lasting immune response, including the generation of memory B cells.
Okay, so we've covered the main principles of adaptive immunity, specificity, memory, and tolerance, and we've talked about how T and B cells are trained to be both effective and safe.
But now we need to talk about the actual weapons of the adaptive immune system,
Antibodies.
the guided missiles of the immune system.
The textbook defines antibodies, also called immunoglobulins, as soluble proteins produced by B cells or plasma cells.
And their job is to bind to specific antigens and neutralize them or mark them for destruction.
So each B cell has these BCRs, which are basically membrane-bound antibodies on its surface.
And when a BCR encounters its matching antigen the B cell engulfs, it processes it and presents it on MHC class II molecules to T helper cells.
And if the T helper cell recognizes the presented antigen, it sends signals that activate the B cell,
causing it to proliferate, differentiate into plasma cells, and produce antibodies.
This whole process takes about 5 days for the first encounter with an antigen. It's called the primary antibody response.
But thanks to memory B cells, the secondary antibody response is much faster and more powerful.
The book says antibodies can work in several ways. They can neutralize pathogens by blocking their ability to bind to host cells.
For example, antibodies in our mucus can prevent the flu virus from infecting our respiratory tract.
They can also neutralize toxins by binding to them and preventing them from causing harm.
And they can opsonize pathogens, which basically means coating them with antibodies.
Making them more appealing to phagocytes, which are cells that engulf and destroy invaders.
It's like putting a big "eat me" sign on the pathogen.
Now, there are 5 main classes of antibodies: IgG, IgM, IgA, IgD, and IgE, and each class has its own unique properties and functions.
IgG is the most common type found in our blood, and it provides long-term protection. It can also activate the complement system, which is another part of the immune system.
IgM is usually the first antibody produced in response to a new infection, and it's really good at clumping antigens together.
IgA is found mainly in mucosal secretions like saliva tears and breast milk, and it helps to protect those areas from infection.
IgE is involved in allergic reactions and also helps to fight parasites.
And IgD is mainly found on the surface of B cells, but its exact function is still not completely understood.
So each antibody class has its own specialized role to play in the immune response.
Now let's talk about the basic structure of an antibody using IgG as an example.
The textbook describes it as a Y-shaped molecule made up of 2 identical heavy chains and 2 identical light chains.
Held together by disulfide bonds, which are strong chemical links.
And at the tips of the Y are the antigen binding sites, which are formed by the variable domains of the heavy and light chains.
So each antibody molecule has 2 identical antigen binding sites.
Making it bivalent, meaning it can bind to 2 antigens at once.
The top part of the Y is called the Fab region, which is the fragment antigen binding region.
And the stem of the Y is called the FC region, which stands for fragment crystallizable region.
The FDC region can interact with receptors on various immune cells, triggering different effector functions.
Now, IgM is special because it's actually a pentamer, meaning it's made up of 5 antibody molecules joined together,
So it has a total of 10 antigen binding sites.
giving it super high avidity or binding strength.
And IgA can form dimers, which are 2 antibody molecules linked together when it's secreted across mucosal surfaces.
And IgE has a unique heavy chain that allows it to bind to mast cells and eosinophils, which are involved in allergic reactions.
So the structure of each antibody class is tailored to its specific function.
Now, when the body first encounters an antigen, the initial antibody response is mostly IgM.
But later on, the B cells can switch to producing other classes of antibodies like IgG or IgA.
This is called class switching, and it's driven by signals from T helper cells.
So the type of antibody produced depends on the type of pathogen and the location of the infection.
Okay, so now the million-dollar question. How do these antibodies recognize and bind to such a huge variety of antigens?
How do we have enough antibodies to fight off all the different pathogens out there?
Well, the textbook says it comes down to the incredible diversity of the antigen binding sites, which are formed by the variable domains of the heavy and light chains.
And within these variable domains are even more specialized regions called complementarity determining regions, or CDRs.
And the most important one is CDR3, which makes the most direct contact with the antigen.
The CDRs are like the fingertips of the antibody molecule, feeling out the unique shape of the antigen.
And to create this amazing diversity, the body uses some pretty clever genetic tricks,
The first one is somatic recombination.
which is a process of shuffling and rejoining gene segments.
So the genes that encode antibodies are not present as complete genes in our DNA. Instead, they're organized into segments, V, D, and J for the heavy chain, and V and J for the light chain.
And during B-cell development, these segments are randomly rearranged and joined together to create a unique antibody gene.
It's like a mix and match system where you can create millions of different combinations.
And the CDR3 region is actually encoded by the junction between these gene segments.
So even small changes at the junctions can create a huge difference in the antigen binding site.
Then there's random reassortment of heavy and light chains, meaning any heavy chain can pair with any light chain.
Further increasing the diversity.
And the joining process itself is often imprecise, sometimes adding or deleting a few nucleotides at the junctions.
This is called junctional diversity, and it adds even more variation to the antigen binding sites.
The book also mentions allelic exclusion, which ensures that each B cell only uses the immunoglobulin genes from one of its 2 parental chromosomes.
So it only produces antibodies with one specific specificity.
And finally, there's somatic hypermutation, which is a process of mutation that occurs in the antibody genes after a B cell has been activated.
This leads to affinity maturation, where B cells that produce antibodies with higher affinity for the antigen are selected to survive and proliferate.
It's like the immune system is constantly fine-tuning its weapons to become more effective.
And all of these mechanisms together can generate an estimated 10 to the power of 18 different antibody specificities.
That's a truly mind-blowing number. It's more than the number of stars in the Milky Way galaxy.
So we have a huge army of antibodies ready to fight off any invader.
Now, how do antibodies and T cells know which cells to target and which ones to leave alone?
That's where the major histocompatibility complex, or MHC, comes in.
The textbook describes it as a set of genes that encode cell surface proteins, whose job is to present antigens to T cells.
It's like a molecular billboard that shows the T cells what's going on inside the cell.
In humans, MHC proteins are also called human leukocyte antigens, or HLAs.
And there are 2 main classes of MHC molecules, MHC class I and MHC class II.
MHC class I proteins are found on almost all nucleated cells in the body.
And they present peptides that are derived from proteins that are made inside the cell.
So they basically show the T cells a sample of what the cell is producing.
And if the cell is infected with a virus or has become cancerous, the MHC class I molecules will present viral or tumor-specific peptides.
And this will trigger a response from cytotoxic T cells, or CTLs, which are specialized T cells that kill infected or cancerous cells.
So MHC class I is like an alarm system that alerts the immune system to internal threats.
Now, MHC class II proteins are found mainly on antigen-presenting cells, or APCs.
Which are specialized immune cells like macrophages, dendritic cells, and B cells.
And MHC class II molecules present peptides that are derived from proteins that have been taken up from outside the cell.
So they show the T cells what the APC has encountered in the environment.
And this triggers a response from T helper cells which are specialized T cells that coordinate the immune response.
So MHC class II is like a surveillance system that eludes the immune system to external threats.
Structurally, MHC class Ig molecules are composed of a larger alpha chain and a smaller protein called beta-2 microglobulin.
And they form a peptide-binding groove that can hold short peptides about 8 to 11 amino acids long.
MHC class II molecules are made up of an alpha chain and a beta chain.
And their peptide-binding groove can hold longer peptides, about 10 to 20 amino acids long.
But both types of MHC molecules only go to the cell surface when they're bound to a peptide.
It's like they need to be displaying something to be recognized by T cells.
And the process of getting those peptides loaded onto MHC molecules is pretty complex.
For MHC class, proteins that are made inside the cell are broken down by a machine called the proteasome.
And the resulting peptide fragments are transported into the endoplasmic reticulum, or ER, which is a cellular compartment.
Where they can bind to newly made MHC class I molecules.
And the complex then travels to the cell surface.
For MHC class II, antigens that are taken up from outside the cell are broken down in lysosomes,
which are acidic compartments inside the cell.
And MHC class II molecules are made in the ER, but they're associated with a protein called the invariant chain, which prevents them from binding peptides prematurely.
Then the complex travels to the lysosomes where the invariant chain is degraded and the MHC class II molecule can bind to the processed peptides.
And then the complex travels to the cell surface.
So it's a very intricate process involving multiple steps and cellular compartments.
But it ensures that the right peptides are presented on the right MHC molecules.
Now, T cells also have these co-receptors called CD8 and CD4 that help them to recognize MHC molecules.
CD8 is found on CTLs, and it binds to MHC class I molecules.
While CD4 is found on T helper cells, and it binds to MHC class II molecules.
These co-receptors help to stabilize the interaction between the TCR and the MHC peptide complex.
And they also play a role in activating the T cell. Now, one fascinating thing about the MHC is that it's incredibly diverse.
There are literally thousands of different alleles or versions of MHC genes in the human population.
This is called polymorphism, and it's one of the main reasons why tissue transplantation is so difficult.
Because our immune systems recognize the different MHC molecules on the transplanted tissue as foreign.
And they mount an attack, which is called rejection.
So finding a good MHC match between donor and recipient is crucial for successful transplantation.
And we also have multiple MHC genes within a single individual.
This is called polygyny, and it adds to the overall diversity of MHC molecules that we express.
So each of us has a unique set of MHC molecules, which helps to ensure that we can present a wide range of peptides from different pathogens.
It's like having a diverse army of MHC molecules ready to display any antigen that comes along.
And this is what makes our adaptive immune system so effective at fighting off infections.
It's a truly remarkable system.
Okay, so now that we understand how antigens are presented to T cells, let's talk about the T cell receptor, or TCR.
The TCR is the T cell's weapon of choice. It's what allows it to recognize specific antigens presented on MHC molecules.
And just like antibodies, TCRs are incredibly diverse.
They're made up of two chains, an alpha chain and a beta chain, each with a variable domain and a constant domain.
And the variable domains form the antigen binding site, which also contains those CDRs.
But unlike antibodies, TCRs can only recognize peptides when they're bound to MHC molecules.
This is called MHC restriction.
And it ensures that T cells are only activated when they encounter a foreign peptide presented on a self-MHC molecule.
The TCR interacts with the MHC peptide complex in a very specific way.
The CDR3 regions of both the alpha and beta chains make contact with the peptide,
while the CDR1 and CDR2 regions mainly interact with the MHC molecule.
So it's a three-way interaction that triggers T cell activation.
And just like with antibodies, the diversity of TCRs is generated through somatic recombination.
But TCRs don't undergo somatic hypermutation.
So their diversity is mostly determined by the initial gene rearrangement process.
But even without somatic hypermutation, the potential diversity of TCRs is huge.
The book estimates it to be around 10 to the power 15.
That's a lot of different TCRs.
Now, TCR engagement with the MHC peptide complex isn't always enough to fully activate a T cell.
They usually require a second signal called co-stimulation.
One example is the interaction between B7 on APCs and CD28 on T cells.
If a T cell recognizes its antigen but doesn't receive the second signal, it becomes anergized.
Which means it's turned off and can't respond to that antigen anymore.
This is another important mechanism for tolerance,
making sure that T cells don't attack self-antigens.
The two-signal requirement for T-cell activation is a crucial safeguard against autoimmunity.
It's like a double-check system that ensures that only the right T cells are activated at the right time.
And the book points out that antibodies, TCRs, and MHC molecules all belong to a family of proteins called the immunoglobulin superfamily,
They share some common structural features,
particularly in their constant domains.
which provide structural integrity and serve as attachment points for other molecules.
So even though they have different functions, they have a common evolutionary origin.
Now let's talk about the different types of T cells.
There are two main subsets, cytotoxic T cells, or CTLs, and T helper cells.
CTLs are the killers. They recognize antigens presented on MHC class I molecules, and they kill infected or cancerous cells.
They release toxic proteins like perforin and granzyme that induce apoptosis in the target cell.
They're like the special forces of the immune system.
And T helper cells are the coordinators. They recognize antigens presented on MHC class II molecules, and they release cytokines that activate other immune cells.
There are several different subsets of T helper cells, each with its own unique set of cytokines and functions.
Th1 cells are important for cell-mediated immunity against intracellular pathogens.
They activate macrophages and promote inflammation.
Th2 cells are important for humoral immunity or antibody-mediated immunity.
They help B cells to produce antibodies.
Th17 cells are involved in the early defense against pathogens at barrier surfaces like the skin and mucous membranes.
They recruit neutrophils, which are another type of white blood cell, to the site of infection.
And T regulatory cells, or TREGs, are the peacekeepers. They suppress the immune response and help to prevent autoimmunity.
So the different T cell subsets work together to mount a coordinated and effective immune response.
And that's how our adaptive immune system protects us from a wide range of threats.
Wow, we've covered a lot of ground today.
It's amazing how much complexity there is in this system.
But we've managed to break it down and understand the key principles of adaptive immunity.
Specificity, memory tolerance.
The roles of B and T cells, antibodies, and MHC molecules.
And the intricate interactions between all of these components.
It's been a fascinating journey.
And we hope you've learned something new about your incredible immune system.
Definitely, and if you're interested in learning more, there's a whole world of information out there.
You can delve into specific diseases or research the latest advances in immunotherapy.
So keep exploring, stay curious, and until next time, take care of yourselves and your amazing immune systems.
See you next time.
Bye.